Hi, you're watching DNA Today, a multi-award winning genetics podcast where we explore everything to do with genetics, from CRISPR to rare diseases to new research. We have won the Science and Medicine Podcast Award for many years now. We have hundreds of episodes, and we really hope you enjoy these conversations where we dive into so many genetic concepts. I'm Kira Deneen, a certified genetic counselor, and your host. In this episode, we're exploring Ambry Genetics' groundbreaking Patient for Life proactive Exome Reanalysis program, and we're going to break down exactly what that means. This unique initiative offers lifetime support to patients with rare and undiagnosed conditions, and they're continuously updating their exome testing findings. So we're looking at those new scientific discoveries to understand how that information might change. In order for me to learn a lot more about this, and you guys as listeners, we are joined by two distinguished experts from Ambry Genetics who are going to shed light on this innovative program and its impact on patient care, because I think this is so important to be updating the information we have. And it's a concept that if you're a longtime listener, I talk about a lot on here of how often should we be looking back at that genetic information to say, okay, how are we analyzing these variants? How are we classifying them? How does this change management and care and diagnoses? So I'm really excited, as you guys can tell, about this episode. So first, we have Dr. Elizabeth Chao, who is the Chief Medical Officer at Ambry Genetics. And alongside her here is Kelly Hagman, who is the Vice President of Medical Affairs and a seasoned genetic counselor. Liz and Kelly, thank you so much for coming on. I'm really excited to be talking about exomes and specifically, as we said, this Patient for Life program where you guys are circling back and saying, okay, we did this a while ago. What's different? What could change? So I figured we should start with, that was my overview of the program, but I don't work for Ambry. You guys have a much better overview. So let's learn a little bit more just about the Patient for Life program and what inspired its creation.
Yeah, thank you. Thank you, Kira. I can take that one. So we have, when you do exome sequencing, these are children that are born with rare diseases. Almost all of the patients that we get in-house are very likely to be some sort of genetic mutation that's causing them. However, today, when we test these children, there's about a 25% chance that we'll find the answer. Why is that? Today, we know about, from all the genes in the genome, we sequence all 20,000 genes in the genome when we do an exome. However, we only know what about a third of those genes do. And we're still learning all the time what the rest of those genes do. In fact, every other day, there's a new research article that comes out in the peer-reviewed literature describing a new gene disease association. And so that means that we now know we have new information about genes all the time that we didn't know before. And so all these patients that we've been testing over the past 10 years of exome, 12 years now, can benefit from this new information. So we actually have a team of scientists at Ambry, 7 or 8 PhD scientists. That's all they do is scour the literature every day for these new papers. And they review them. We have a scoring system that we use, very similar to like a scoring system for variant classification. And they score the evidence of this new gene disease association. And if it reaches what we call characterization, we then are able to go back to our database of all patients that have ever tested at Ambry for exome that had a mutation in that gene. And we can say, yep, this one matches this new phenotype that's being described. And we can then send out a reclassification report from negative to positive for those patients. This started because maybe about 2 years into offering exome sequencing, we had a client, a very savvy genetic counselor, called up, and we used to put in the back of the report a bunch of like uncertain findings and genes that we didn't know much about. And she was on her lunch break reading through the American Journal of Human Genetics.
As we all do.
As we all do. Yes. So she's a gem. And she recognized a name of a gene, and she recognized it from her patient. She thought, this actually sounds like one of my patients. And so she called me and she said, I think my patient has this. And I looked back in the report. Sure enough, we had put it in the back in the reference list. And then I thought, well, we need to do this. We need to find these. This shouldn't be a clinician. Not all clinicians are going to be reading American Journal of Human Genetics on their lunch break. And they should, most of them don't have a lot of time to do that. And so that inspired what we call the gene team today, the team of scientists that's reviewing the literature all the time so that we can go back and classify patients. And this results in overall 5% of patients over time that had an original negative or uncertain result now have a positive result, and it's ongoing and it continues to increase.
Wow. And that's 1 in 20 patients. Like that's pretty remarkable. And that's just what we have now to be able to do. I think it's as time goes on, we're able to classify more and more variants because we're getting more and more of this data that we've talked about in other episodes where at some point we're probably going to get to a point where VUSs don't exist, hopefully, that we're just like, okay, you look at the literature, this is how we're classifying it, done. I hope to see that in my career. I don't know when, I've heard different number of years from people, but I think that's such an important part. And I think one thing I want to pull out of this is that, and correct me, it sounds like Ambry is just letting the clinicians know that ordered this testing. It's not that, let's say I was a peds genetic counselor, I'm not, I'm prenatal, but if I was and I had a patient 5 years ago, it's not my responsibility to go back and find that. You're going to let me know and be like, hey, remember that patient 5 years ago? Let's check back in because now we have a causative pathogenic variant that we are like, this is what is leading to this disease. Is that fair to say?
Yeah, that's exactly right. Because we want to shift the burden, the place where I think it's best suited, where we have the best chance of finding an answer going forward. And in many cases, that really is the laboratory. Clinicians have their patients, they often follow them for a long time. They're followed in medical genetics clinic. And we do encourage them, reaching out every time, there might be new phenotype information or new family information that might be a good reason to undergo reanalysis. But our data and lots of the data published in the literature really shows that the most powerful tool we have for reclassifying these cases is understanding new gene-disease relationships. And that's where most of the benefit from reanalysis comes from. And I think the lab is in the best position to do that. Because as we're curating, those gene-disease relationships every day, we're curating the literature, we're learning more. It's not just about that one patient, that a medical geneticist or genetic counselor might be keeping track of. It's about all of the patients that we've ever sequenced. So like in Kelly's example, we made a diagnosis in that one patient, but we also have the opportunity to go back into our database and say, hey, what other patients did we identify a VUS that we identified a suspicious variant? And maybe we can diagnose those too. And so that really is a lot of the power of Patient for Life is we are helping all patients who've had testing at Ambry very, very broadly and not just the one that might have reached out and requested reanalysis at exactly the right time. We're doing it proactively, and we're shifting that burden into the lab where I think we can do a really good job. We do a really good job at doing this sort of reanalysis work.
And there's a lot of steps that go into this. Kelly, you were talking about some of them in terms of having is it the gene team that is looking through all this data, right? And so of them just scouring through and, okay, what are these new papers? Is this enough evidence really to say, all right, this genetic change really is causative pathogenic? Are there other steps involved that you wanted to highlight in terms of how this process works? And also how often it's done? You're talking about your team just, continuously working on this, but just in terms of maybe how often it is that something is found that you're like, okay, now we're reaching out. Is this something that happens a couple times a year, a couple times a day? Like what is that frequency?
There are several steps involved in analyzing a patient's the result and in analyzing the literature for new gene disease associations. I described the gene team previously, and they're every day looking at the literature for new gene disease relationship papers that are coming out. And there's a lot of them, and not all of them actually provide enough evidence to reach characterization for a gene, but it goes into our database and we're able to say, okay, there's additional points at this point. And then if another paper comes out, maybe we'll eventually achieve more points. Because we were committed to analyzing gene disease validity, we published a scheme for classifying genes. It's very similar to a variant scheme. However, this is for gathering enough evidence to show that a gene is associated with a particular disease. To date, we're the only laboratory that has published a scheme about gene characterization. And we use this scheme, we're updated all the time, just like we do our variant scheme with new evidence to make sure that we have the most up-to-date information. And so The process is very laborious to do this, to do Patient for Life, but we're committed to it because we want to find an answer for every single patient. It drives us crazy when we have a patient that has something going on and we just didn't find it. And knowing that we have the Patient for Life program, we're like, okay, we're going to keep looking forever until we find the answer for this patient. And so the gene team starts by identifying the literature. Whenever they do score a gene disease relationship high enough to reach characterization, they alert the reporting team. And then the reporting team then goes into the database and looks back through all the past patients that ever had a variant in that gene. And they'll look to see, yes, this one looks like it's a good enough association. This one's not. And then the ones that were, they'll issue a proactive reclassification report. They alert our genomic science liaison team who interface with clinicians very closely. And they'll call the clinician and let them know, because sometimes it's been years since they sent in the sample, to let them know that we have this new finding. Sometimes we'll need a new sample so we can confirm in the laboratory. That's pretty rare these days, but sometimes we do. And so they work with their genomic science liaison to communicate about this new update. And they're generally all very excited, very pleased that we're able to find an answer for these patients.
And you've had a lot of success stories in just what you were sharing of being able to have that first genetic counselor that was like, hey, I figured this out. And you're like, we should start doing this. As many programs and things start by someone having an idea and then, that grows. Are there any kind of types of success stories, keeping HIPAA in mind of protecting patient confidentiality, of maybe like common findings that then we were able to find a cause to that? Anything coming to mind?
Yes, we have so many examples to draw upon, but one that comes to mind is a patient that was an infant, a 5-week-old male patient that had abnormal brain MRI, irritability, elevated lactate. He also had an affected brother that had passed away at 2 months of age, who had the same clinical findings. They ordered a rapid exome for this patient and it was overall negative, unfortunately. And then the very next year, a paper came out on the gene HPDL, which had the same overlapping symptoms as our patient. We scored the paper and it reached characterization. So then we then went back. We were able to issue a positive report for this patient and 8 other patients that had previously been tested that were either negative or uncertain. And we were able to reclassify for them as well. So, and this is just one example of many that we do on a regular basis. And again, this is not, I just want to emphasize this because, I know how important it is for parents to find an answer for their children. They want to know why this happened. The very first time we ever got a positive exome result was on a patient that had been waiting 15 years for her sons to get results. And we found the answer for them after 15 years. They tested everything under the sun. And the mother burst into tears when she got the results and just said, I finally know this wasn't my fault. And I mean, it still gives me goosebumps today when I tell that story, but this is what happens every time we issue a positive result. So that's front of mind of everybody in our lab, which is why we're so committed to finding an answer for everybody. And so this is why we're not stopping until we find an answer for every single patient. And this Patient for Life program, it's not happening in any other laboratory. They don't have, and I say that because I just think it's so important to use a laboratory that is committed to finding the answer for patients. So these 8 patients would very likely still be negative or uncertain in any other laboratory unless they were diligent enough to find the paper and go back and find their own patient's result and ask for a reanalysis. This is taking the onus off of, to Dr. Chao's point, this is taking the onus off of the clinician and putting it in the laboratory where it should be. And we're here to find the answer for you. You sent us the sample. We're now committed to finding that answer for you.
And that's so important because as a genetic counselor that works in the clinic, I don't have time. I only have so much time to even just finish my patient letters, make sure all my requisitions are complete, the results have been called out, I'm prepping for the next day. I don't have hours to sift through of looking at, oh yeah, this patient I had a year ago, and even to have them in front of me, so it's just, it's so helpful. And from my perspective, I feel like it should be on the lab side and responsibility to be updating this, because to put that on the clinician's plate, even for outside of genetic counselors, that's even harder, right? Because they're not even necessarily ordering genetic testing all the time, other types of providers. So to me, this just makes sense of like how the workflow should be, and that the labs are the ones that have the team members to also know how to sift through and understand, as you said, of classifying these variants and how much evidence do we need. I have not been trained in that, right? I can read a report, I can call the lab and understand more and ask pointed questions, but I don't know the true steps that need to happen to truly classify a variant with what the information we have. I could give it my best shot, and it's something, right? Better than, maybe someone that doesn't have a genetics degree. But it's so important to have that on the lab side. And it's surprising that other labs don't have this, but I'm aware of that. That Ambry is really a leader in this space. And with that, I think it's important to understand how Ambry ensures the accuracy and reliability of these gene disease validity assessments, because there are times where there is a correlation but not causation, and figuring out the difference between those. And genetics is just really complicated. So what is some of what goes into making sure that that's the case? I mean, Kelly, you talked about the process, I guess, of how Ambry classifies variants, and that's going to be different depending on the different labs, which I think is an interesting concept that deserves its own episode. But what else goes into this that maybe we haven't talked about yet?
So, I mean, I think, as you suggest, there's a framework for classifying variants. And then there's also a framework that we built for classifying genes. And I think that's where you can depend, on the robustness of the process, the fact that, we have a validated framework, we've put it together. A lot of the work that we've done is similar to work that's happening, also with collaborative groups. For example, at ClinGen, they built out a process also for assessing gene disease validity and have, biocurators who are working on that as well. We've, internalized a similar process. We've optimized it. We sort of dedicated to our own process, the way our own databases are set up, to ingest that information and, pull the most that we possibly can out of it in terms of finding answers for parents, for patients, and for clinicians. Because as Kelly alluded to, the laboratory is in the best situation. We know how to assess gene disease relationships. We know the difference between, as you said, correlation, causation, how to put that into a validated framework to say that a relationship is definitive. Is it strong? Is it moderate? And where that becomes something that should be clinically actionable. Similarly to how we do for variants, we call them uncertain, we call them likely pathogenic or likely benign. The same framework exists for us for genes. And that's where the majority of these results come from, is as we build on those relationships. I think one of the really unique things about exome or genomic testing is that it changes over time, right? So even though we've been offering, exome testing since 2011, the test that we offer today is better than the test that we offered yesterday. And yet it's not as good as the test that will be available tomorrow. And so with something like Patient for Life, you really get the test that's not just available when it's ordered, but the test that's also available next week, next month, next year. As the science moves forward, we're going back to your results. We don't need to resequence your exome because the data remains the same. It's stable. That's your germline exome. We know what the letters are. We know what the bases are in the sequence. But how we interpret it changes over time. And so this allows us to take advantage of that in a really robust way that, we believe clinicians and families can depend on to make today's test as good as tomorrow's is going to be, and keep everyone on that highest level of quality.
And that's something that's so unique in genetics compared to other areas of testing in healthcare, that you do a test and we have this information, we can keep revisiting the information, whereas in other kinds of tests, we need a new sample. So that's something that is just very unique to genetics, that we can keep looking back at this, and without the patient even necessarily being aware that this is being worked on for their benefit, and this is continuously being updated. There's also, as with all genetic tests, ethical concerns to have with this, and logistics too, of, all right, you reach out to someone 5 years later, that clinician may not be working in that position anymore. They may not have contact to that patient. Phone numbers might have changed, email addresses, physical addresses of where they live. So what are some things you guys have to take into consideration, especially on the ethics side when you're reanalyzing exomes, especially as the years go on and on?
The logistics are a huge challenge, and we've actually worked the logistical approach, or the ethics of it is really a twofold question. One, the laboratory finds something new, we have to reach out to the healthcare provider. Then once we've contacted the healthcare provider, they of course have to find the patient and family, and so it is a challenge on both ends of it. We actually worked closely. I was involved with an effort through ACMG a few years ago to put forward some considerations for both the laboratory and the clinician in reanalysis and reclassification, just some points to consider, in both cases, to think about some of those issues, to think about confidentiality, to think about the original consenting process. I can speak to our processes as well at Ambry. We have processes in place. We don't simply, I don't know if anyone faxes anything anymore, but we don't simply fax out a report. It's the last known fax number that we have from back in 2015, right? We do sometimes have updated clinician information. We always make a point of contacting the clinician first before we send out any private health information. We reach out, we make sure we've connected with the right provider or the right institution where a provider may not be, but we know who's taken on their practice and their caseload. And we make that connection before we send out any of that information. But we do understand and keep in mind that really is only step one, because then the clinician's going to go through that process, making sure that if they're not still seeing the patient regularly, they have to then transmit that information appropriately to the patient.
And are there cases where you can't reach out to that clinician or that institution or whatever, and you're in this awkward position where you're like, we have information that the patient really deserves and should know? Does Ambry say, okay, well, maybe we should reach out directly to the patient when all efforts have been exhausted with reaching out to their initial clinician?
Yeah, that's a good question. We don't reach out to patients even at sort of the end of that process. But I do think that's an important question to keep asking in terms of the ethical and legal considerations around that. We typically actually don't often we take patient information as it relates to billing and some other aspects of the test, but we often don't even have as much patient-specific information as we do for the ordering provider and the clinic that we're working with. And we're usually successful in tracking down, the clinic or whoever's taken off that practice. But I can imagine that there are situations where that might be appropriate. I think it just needs to be defined, some of the parameters around that, where it might be appropriate for a lab to actually reach out directly to a patient.
Yeah, that makes sense. In today's day and age, I mean, usually it's pretty easy, email, whatever. It's like there's usually one way, whereas like, back before email and cell phones and all that, a little bit different, right? And I think when it comes to talking about going through data like we have been and being able to give patients a diagnosis, and just, Kelly, that you were sharing of how useful that is, but also just emotionally too, of finally having an answer. I think for people that have been on a diagnostic odyssey for so long, it is so important. And I would be remiss if I didn't bring up the disparities that are present, that we started the field of genetics looking at mostly European genomes. And so we have a lot of data when it comes to classifying variants for people that have recent European ancestry. But for other types of ancestry, as a genetic field, we are lacking. We do not have an equivalent amount. And so there then is this downstream effect that people that don't have recent European ancestry are not getting the same level of diagnoses and having those variants interpreted correctly because we don't have enough data. Within Ambry, in this program, the Patient for Life program, how do you guys address this just in terms of different genetic ethnicities to try to level that, knowing that this is a problem in our field? But we all have to start tackling this. Each lab, I'm putting it on you guys to help tackle this, along with the big, huge, genome projects and different things out of NIH and other countries.
Yeah, and we still do see that across the board, across all tests, not just exome. We still see a higher rate of VUSs in non-white patients. And this is just kind of an artifact of, as you mentioned, our database that have been predominantly white patient or white volunteers that are providing their data, and where research has been done. However, one of the great unintended consequences from Patient for Life is that we're finding that that non-whites are actually significantly less likely to have a provider request a reanalysis, like to have the provider ask for a reanalysis. However, they're actually significantly more likely to have a reclassification through Patient for Life or through a reanalysis request compared to whites. So we're finding that the Patient for Life program actually in itself is helping reduce these disparities. So it's not on the onus of the clinician to ask for a reanalysis. They have to go back and kind of systematically find the time to request these. We're doing that for them. And then, yes, we definitely have more non-whites that are getting reclassified through this program and through reanalysis requests.
That is such a light within this dark disparity, because that makes sense. The variants that we're finding over time are more likely to be non-European, non-white, because that's the newer information that we have been lacking before. I don't know why I didn't piece that together, but when you said that, I was like, light bulb, of course. Like, people that are of different ethnicities are going to benefit more so, because that's the data that we're able to gather and analyze in that time. And then also when you were talking, I was thinking, I'm thinking, okay, Ambry's going through and updating and looking and reanalyzing these exomes. But yeah, as a clinician, you could reach out and be like, hey, I have some new information maybe about a patient, that they have a new symptom or something, and I want to let you know, because maybe that affects how you're analyzing the exome. Do you guys have a setup for people to be able to reach out, healthcare providers, and say, hey, here's some new information that might help with this process?
Yeah, we do. Of course, provider-initiated reanalysis, we do have a system where they can reach out, and phenotype information or new family history information are probably the two most powerful tools there. So oftentimes, we might do the initial analysis, for example, on a newborn where there just wasn't an opportunity. There might be, congenital anomalies, but we don't yet know anything about the development or, many other aspects, many other features that will develop over time. Sometimes, there's another sibling that was born that might be affected with a similar phenotype that gives us a reason to go back and look at specific genes or specific inheritance patterns. And so we do welcome clinicians to reach out to us when they have that type of pertinent information available. In terms of our reclassification processes, it hasn't proved to be as useful as what we described in Patient for Life, but it is of tremendous value, and we still strongly encourage it, because once we have that information in our database, it's going to then be part of the Patient for Life program. So when we go back and we're looking at all those genes, if we know, that there were, these specific developmental delays or these specific features that develop later on, that's going to really drive our ability to be even able to do a better job at reclassification across the board.
Yeah, that's so exciting. This is so needed in the field, and it is fantastic that Ambry's really leading the way with this, of being right now, as we're recording, the only lab that I'm aware of, and you both have echoed this, that is offering this reanalysis exome program, and just how much success you've already seen with this. So I'm really, really tuned in to see just the progression of this program and how helpful it is. And I'm imagining then other companies, copying this format and saying, hey, Ambry really showed us that this is the way and a service we should be offering as a laboratory, and for this to be standard with labs. And then Ambry being credited with, hey, we started that, so we started that trend. But thank you both so much for coming on and explaining so much of this to me. I think this is something that I've kind of been referencing in a lot of episodes of, hey, and going back to the original sequencing and seeing what's new. And something that I talk about with patients a lot too, just, even within my role of in patient-facing care. So thank you so much. I think this has just been really enlightening, and I can't wait to, have you guys back on and be like, all right, so what are the updates? Like, what are the big changes that you've seen in terms of just all this? And one day being like, VUS, what's that, right? Of when students will never have heard the term, and it's just in our old textbooks. But thank you both so much. I really appreciate you coming on and sharing all of this.
Thank you. It was a pleasure to be here and to be able to talk about the work that we're doing.
Thanks for watching DNA Today. To access all of our episodes, head over to dnatoday.com. We also have a lot of bonus content on there that you can enjoy. If you have any questions, comments, suggestions, guest pitches, you name it, send them in to info@dnatoday.com. We'd also really appreciate if you could take a moment to rate and review the podcast on Apple, Spotify, wherever you listen to the show. It really helps more nerds like yourself find the show. Also, if you like giveaways and other ways to connect with us, I recommend following us on social. We're at DNA Today Podcast. We also have a Patreon if you want to be the most level involved in the show. That's also at DNA Today Podcast. Thank you so much for listening and watching. Join us next time to learn and discover new advances in the world of genetics.